on hemp variety selection and regulations. We're here with a great group of panelists, some breeders, researchers, and industry players. And we're gonna dive into some of the genetics and regulations and some of the things that might be keeping some folks up at night here that are participating. So today we have Jason Evans from Tweedle Farms, Chris Hone from Kyogene, Dr. Daniel Putnam, PhD from UC Davis, and Dr. Bob Hutmacher from UC Davis as well. And again, this is hosted by myself, George Workman at Kola Seed. And we're gonna dive into each of the panelists and we'll have everyone give a brief introduction to themselves. Just a quick background on the webinar itself. We're looking to really give folks an insight on what distributors like Tweedle Farms are looking for in hemp products when they retail them, what the regulatory bodies are looking for with these upcoming years with these new regulations. And we're also going to talk about the importance of genetic selection and how breeding is evolving to meet the needs of both the regulatory bodies and the retailers and consumers. My name is George Workman. I'll be hosting here. I run Kola Seed. We're a seed distribution company located in Central California. We work with established breeders, researchers, and other supply chain partners to provide good genetics and learn and grow the hemp industry, the cannabinoid-based hemp industry. And in doing so, we've worked very closely with Jason Evans at Tweedle Farms, and we'll start with his introduction. Jason, I'll let you take it from here.
Thanks, George. Thanks for having me. Yeah, my name's Jason Evans. I'm the co-founder and co-owner of Tweedle Farms. We are a registered hemp farm up in Northwest Oregon. We also own and operate the retail website, tweedlefarms.com. We have been growing hemp in Oregon actually 1 year since the 2014 Farm Bill started. So this is our 7th year growing hemp in Oregon. We grow primarily for the smokable flower market, and we also extract for ancillary products, topicals, edibles, and other inhalables. It's great to be here.
Thanks, Jason. Next, we have Chris Hahn from Kyogene.
Yep, thanks everyone for joining us. So I'm the Director of R&D at Kyogene, leading breeding and breeding technology development for the company. And we're focused on breeding new varieties to meet the needs of growers and the end consumer in the hemp industry. And I'm glad to be here today. I want to share with you guys a little bit of my perspective on hemp breeding.
Thanks, Chris. And finally, we have Bob Hutmacher and Daniel Putnam. We'll start with you, Bob, and then go in and have Dan introduce yourselves.
Okay, thank you. I appreciate being included in this. I'm a cooperative extension agronomy specialist with the University of California and located down in the San Joaquin Valley at the Westside Research and Extension Center. And we've been working the last couple of years on industrial hemp, Dan Putnam and I and associates, initially focusing on plant density responses and irrigation. So glad to be included. Thanks.
And Dan, if you could introduce yourself, that'd be great.
Sure. Dan Putnam. I'm here at University of California, Davis, about 4 hours north of where Bob is in Fresno County. And I work primarily with forage crops and alfalfa, but in the last couple of years, we've been actually working with hemp and also, you know, with an interest of making sure we learn more about an introduced crop and are able to work on the agronomic practices of that.
Thanks, guys. Yeah, it's great to have a diverse panel here today to get different insights from different sectors of the supply chain. We're going to move along here and dive into an intro based on just a little insight on what we're going to be chatting about. I'll start out with saying some things that are obvious to most, that this hemp market has been a moving target since the beginning. The consumer interest, the demand, what folks are looking for, what's acceptable, what's not, that's been going in and out. Then we've got federal, state, and local regulations. Some counties banning hemp altogether, some states banning hemp altogether. Now I think all 50 states are online, and now you're just getting local ordinances and state ordinances putting rules together on certain things. Some of the trends that we've seen, we all know about, are smokable flower, which seems to be actually sticking. That's one of the trends that's actually kind of held true and is growing year by year. We have CBG, Delta 8, CBDV, the varieties, and other things that folks are doing to pivot to try to keep their productions profitable. And one of the other things we're going to talk about is trial data, and that's one of the things that from a seed perspective and genetics perspective, we're very involved in. We work with UC Davis on that, and Dan and Bob have done just an incredible job the past couple of years in identifying some industry pain points, some of them being irrigation related, agronomic issues, some of them being regulatory issues like this THC compliance. And so they're able to use their lifelong experience of research on crops to generate graphs, data, and things that can help us as growers and supply chain participants understand what we're doing a little bit better, and maybe help us in our decision making to help bring a more profitable, less risky business approach to the table. And finally, we have all this trial data, all these trends, all these consumer interests, and that all leads into variety selection and breeding practices. So Chris is really going to dive into how all these things above are influencing the breeding program, and what the breeding program is doing to adapt and move quickly enough to keep up. And with that, we're going to start with Jason and dive into some of his insights from a producer, a processor, and a shipper. Jason, what do you think are some of the biggest risks that you're seeing? Can you tell some of the listeners, from your perspective, what you've been working on, and how you see this unfolding over the next couple of years in terms of regulations and et cetera?
Yeah, thanks, George. Before you get into risk mitigation, because each sector of the hemp industry has its own, inherent benefits and risks. So for purposes of this conversation, we do not grow for fiber. We do not grow for seed, whether it be for propagation seed or for, edible seed for grains. We grow hemp flower for the smokable market and for extraction. So I'm going to focus on those things. I think the biggest risks, there's a couple. There's internal and external risk. I think for the year coming up, and since the hemp program started, some of the biggest risks outside of weather and keeping your crop healthy and happy are in the legal framework, primarily with THC concentration, because you could have the best soil and the healthiest plants in the world, and if it doesn't pass muster with your respective Department of Agriculture and now the USDA, you can't monetize your crop, and you could potentially see federal criminal charges depending upon the THC concentration. So I think I know now that USDA has come online and their interim final rules took effect March 22nd. Point 3 is the law of the land, pre-harvest and post-harvest, especially if you're growing flower for the smokable market. 0.1 up to 0.1, or excuse me, 1.0, that's where you start getting federal criminal charges. But anything above 0.3, you're either going to have to remediate or destroy. At least the USDA's given us a pathway to remediation and destruction of crops without having law enforcement involved. Between 0.3 and 0.5.
And Jason, Tweedle Farms is a online retailer of hemp flower, correct? TweedleFarms.com.
Yes, sir. We started off growing primarily for the extraction market, but then we also saw, coming from California and being in and around the cannabis industry for most of my adult life, we kind of saw a hole in the market where, it's ultimately it's all just cannabis, whether it expresses, THC as a primary cannabinoid or CBD as a primary cannabinoid, or now CBG as well. People like to smoke cannabis. And at the time when I built the website, there were only a half a dozen vendors online, and they were all based out of Las Vegas or Miami or Fort Lauderdale. And there's nothing wrong with those areas per se, but those were not areas that were known for their cannabis production. They were just online companies only. So we saw a hole there, and we were the first farm to table raw hemp flower provider in the country.
Would you what desired genetic traits and retail attributes are you looking for when you sell on your platform? Well,
to take a step back again, depending upon what you're growing for. If you're growing for fiber or seed, you're going to have a different growing method. You're going to have a different type of plant, different cultivar that you're going to select. For this, if you're growing for cannabinoids and you're growing for retail distribution of smokable flower, it's the same 5 things. You want as high a cannabinoid and terpene profile as possible, albeit within USDA 0.3 total T compliance. You want good bud density, good bud structure. Coloring is getting more and more important to consumers. It's you kind of look at cannabis consumers like wine connoisseurs. They like a good nose, they like a good touch, they like a good feel, they like a good smoke. A lot of the different cultivars out there express their flowers in different ways. So, and it's nice to have a variety as well. But specifically for the retail market, bud density, bud structure, cannabinoid content, and I think what's not been given enough credit, and what we're going to be focusing on heavily going forward, because we pivoted to total THC at the end of 2019, so it's not an issue for us like it'll be for a lot of other farmers, is cannabinoid to terpene ratios. Everybody understands 28:1 CBD to THC ratio in raising hemp, but nobody's really focused on cannabinoid to terpene ratios. And especially when you start considering the potential efficacy of different terpenes in conjunction with cannabinoids in that entourage effect. Terpenes are just as important as cannabinoids. So that's, that it's, you have to have good legs. You have to have good, dense buds that smell and have a decent amount of cannabinoids on it. People are going, the retail consumer is going to more and more become more educated. And as more cannabis platforms come online, whether either, in the marijuana world, in medical and or adult use programs throughout the states, they're going to get used to more dispensary quality flower. And so that will become the expectation going forward.
That's awesome. That's such great insight. And it'd be great to have you chime in again here when we talk about sampling down the road, when we bring in Dan, and I know you have some thoughts on the USDA regs that Chris is going to chat about as well. So as we move along, what we're going to look at here are some regulatory highlights, and I'm going to really bring Chris into the presentation. He's been very well studied on this. He's read up on it pretty closely, and he's just going to share a few highlights that he sees as, you know, the big points in all this. And and I think Jason will chime in as to, you know, what he sees as you know, his big points as well.
Yeah, so I just wanted to take some information that you can find actually directly on the USDA website and throw it out there. These are high level kind of bullet points that they highlight on their website. And the main point being that these things will be different depending upon the state that you're in, because the federal government, the USDA, has set the general regulations, and then the states are deciding how they're going to implement that, and they could be more strict, of course, just like with any regulatory structure. So it's going to be dependent upon where you're at, and you always need to check with your local governments. But these are kind of where the loosest that it can get. So it can only get more stringent from these points based upon the local government. So one of the main things is the negligence, which I know Jason touched on, and I'm sure he'll chime in a little bit here. It's set at 1%, but as mentioned, this is where it becomes negligent. It doesn't mean that you're compliant if you're below the 1%. 0.3 is still the rule. When you do your pre-harvest test, this is mainly where this negligence is of concern, because
it's a criminal charge, right? Am I right in understanding that?
Yeah. So yeah, if you do your pre-harvest tests and it tests below 1%, but above 0.3%, then there's some courses of action that can occur. And I'll let Jason explain that a little. He knows that a little bit more than I do. But that's not where the concern ends, because yes, that's the first concern of the farmer. Can I harvest my field? So you do your pre-harvest test, and then it says, okay, now you can harvest. But we all know that from that point, cannabinoids continue. They don't stop after you've tested. So up until the day you cut the plant and take that out of the field, cannabinoids are going to continue to increase. So if you're like Jason and you're shipping across state lines, you have to consider that if you harvest too far beyond the point of maturity where the plant has gone beyond 0.3%, now you're going to get into situations where you could get federal repercussions, especially if you're above the 1%. Then it's, you know, becomes criminal charges. So that's one of the main things. I wanted to also just point out that the USDA has kind of given it to the states to look at disposal and remediation options for non-compliant plants. So it allows for alternative disposal methods for non-compliant plants, which don't require law enforcement. But this depends on the state. In California, they still don't know exactly what they're going to be doing. I talk to the Ag Commissioner's office almost every day, but it goes back and forth. So it's still a moving target, just like George mentioned. But hopefully things settle in now that the USDA final rule is there. California did some things to align with what they thought the USDA was going to be doing in terms of the timing of sampling, which was 15 days, or it was 14 days. And so California switched to that. But then the USDA said, oh, okay, well, after stakeholder feedback, we are going to move it to 30 days because that makes more sense. So then California now is trying to shift back to the 30 days, which they were already at. So it's this big mess, but things will settle in hopefully and become more clear for us all as we go. You just got to be talking to the local ag commissioner, day by day, because things are changing that quickly. And then around sampling method, it again allows the states and tribes to adopt kind of their own protocols for what sampling methods are going to be used. And the USDA even outlines that it could be a performance-based approach to where if you have a grower who's growing a certified seed variety, then they might approach that differently than someone who's not growing certified seed. Or they can take into account, well, this grower has been compliant for the past 5 years and we've never had an issue. So we're going to approach this grower a little differently. So that's kind of cool that the USDA has built that in there, but we'll see how the local regulators take to that flexibility. And I'll let Jason chime in if there's anything that I missed or if there's any key points that you wanted to point out to anybody. That's all I have to say.
One quick thing before you get going, Jason, that I noticed from a seed sales perspective is, we had a customer in California has a 30-day testing window. They test the 5 plants. The pre-harvest was no problem. But in Washington last year, we had a customer, they had to test 30 plants and they had to make a composite, and it was 2 weeks prior.
2 inches.
And 2 inches of the bud. So you can see what a big difference that is from California to Washington. Even though we're all governed under the USDA, it's quite a variance there.
Yeah, thanks, guys. So quickly, again, I'll just remind everyone that it depends on what you're growing for. We have been growing under the hemp program since 2015. We have never had one pre-harvest test come back non-compliant. That is primarily due to choosing good seed vendors, choosing reliable seed vendors. If you are growing for fiber and feed, you're going to be talking to a different seed vendor generally than if you're growing for cannabinoid content and flower. So know your vendor, make sure that they're vetted, get certified seed, talk to other people that have bought the seed before. Again, in the last 7 years, we have never had one hot test because of the genetics that we chose. I also recommend that if you're growing for, even if you're growing just for biomass for extraction, or if you're growing hemp for the smokable flower market, that you test early and test often, in addition to the state and federally required pre-harvest tests. Okay, we generally test each one of our varieties, whether we're growing greenhouse, indoor, out in the field, summer, winter. We will start testing for potency at week 4 of flower, because we want to see how the plants are performing, and it also gives us a gauge to just double check to make sure that we're compliant. But we test it.
Can you talk a little bit about the post-harvest aspects of this?
Yeah, absolutely. And let me just jump into these numbers real quick. 0.3 is the number plus or minus a measurement of uncertainty. Okay. In most states, that measurement of uncertainty is going to be between 10 and 20%. So 0.3 could actually mean 0.36. Nobody, I guarantee you, nobody in the state of Oregon is going to give us a problem at 0.34 or under. Most states are falling in line with that. The USDA has not defined their measurement of uncertainty yet, but they will by the fall. Remediation is in most states 0.3 to 0.5. 0.5 to 1 is destruction. Anything over 1% is not only destruction, but a one-time, one slap on the wrist, and then it's criminal charges. Now, that's all pre-harvest, but bear in mind, folks, if you're growing for the smokable flower market, you're going to be drying, trimming, curing, and concentrating the cannabinoid contents across the board. So in the state of Oregon and many other states that have already adopted USDA policies, there are post-harvest batch testing requirements that must be done in addition to a pre-harvest test. The 0.3 still applies there. In most states now that are getting caught up to states like Oregon for post-harvest batch testing, it can't be a sample that just gets turned into a lab. It has to have a lab sampler come out, test, randomly sample and test your batch, and also test, in addition to potency, for pesticides and moisture. We additionally test for terpenes, microbials, and heavy metals on every single batch. And we publish those results on the product page of each single variety we sell. It sets this kind of stuff is expensive, but it sets you apart in the retail market. Again, the public is getting a lot more savvy as to what's good product, what is safe product, and what vendors have been getting away with, that really, if there's no regulatory or law enforcement of these regulations, it's up to the public to choose the best growers and vendors. So,
and I'm sure we're going to have quite a few questions on this too at the end. So I noticed some folks are chiming in via chat. We will get to your questions at the end and plan to do so via the chat function here. So you guys are on it with the right function, just a little bit out of queue in terms of timing. So we'll touch back with you here at the end. As we move along, we're going to jump into breeding considerations. And thank you, Jason, for the insight again.
you bet.
Chris is going to jump in here and jump into what he's getting into in the breeding world. Chris, what are some of the strategies to meet the needs of all the supply chain steps?
Yeah, so when breeding a variety, you have to take into consideration a lot of things that Jason has already mentioned. The number one thing being agronomic traits. So the first concern, the first risk for the grower, who's our main customer, is getting to the finish line. So being able to produce the crop that's going to be healthy, yield well, and make it to the end for the grower, and then also be compliant. And then the grower has to be able to sell that product. So that product then has to meet the needs of a processor. So high cannabinoid content, if it's going for extraction, bud quality, bud density, trichome density, terpenes, and these sorts of things, bag appeal, if it's going into the smokable flower market. So it needs to meet those consumer needs as well. So we have to take all of these things into account when designing our varieties.
And now you have some pretty interesting technologies that are helping you basically identify like a trait, like such as like bud density or like the color purple in the flower, or maybe a large bud amount on the flower with a high cannabinoid concentration for processors. Are you following? Are there any of these modern day tools that you found that really help you with that and help you grow your breeding program larger and help you kind of keep up with the fast-paced nature of the industry?
Yeah, so we're from the beginning, Kaigene's invested heavily into developing genomic tools. Coming out of the seed world, I saw this in my experience with vegetables, and those companies that invest into those technologies, which can advance the breeding program, advance varieties into the market quicker, tend to do the best. So we invested heavily into genomic technology, into, for example, molecular markers, so that we can speed up the breeding process. Because with molecular markers, we can take and utilize our breeding space more effectively and efficiently. So we can take and test seedlings before they ever go into the field or the greenhouse and know some of the phenotype attributes that they'll have. So if they don't check one of the boxes that we know we must have, so if it's a must have within that variety, we can keep it out of the field and keep it out of the greenhouse, take it away from taking up space for us, so that we can then put plants into that space that we know will have at least those must have, so we can evaluate those. And
wow, that must save you a lot of time and money, I bet.
Yeah, so it saves time and money, and lets us be more efficient and move things through the program quicker, so we can make improvements pretty much year to year. Right now, things are just going so fast.
One of the fascinating things that I've seen with Chris's work is the variation in cannabinoids within populations. So for example, when you look at a couple of varieties. I'm just gonna, I don't want to name exact varieties, but competitors' varieties, really common varieties in the marketplace. You'll see that some of them, yeah, they test at 0.6 and 15 THC and 15% CBD, but then you'll find other phenotypes within that same population that might be at 8% and compliant. So just that's another thing we want to tell folks is like, don't look, make sure you ask for a minimum of 3 COAs and don't just let somebody give you the unicorn COA and base your whole program off of that, or base your whole purchasing decision off of that, because it's not so much the fact that hemp is stuck at not being non-compliant, it's the fact that hemp has not been bred for long enough amount of years to isolate these families, isolate these phenotypes, and generate stability around them. And then this is where the genome sequencing, the QTL markers, having a larger breeding program that's more focused will help us get there over time.
Yeah, exactly.
Thanks, Chris. We'll keep moving it along here. And this is basically comes back to the importance of the research and variety trials, where we're going to really loop Dan and Bob in, some of the researchers from UC Davis. They've done some incredible studies on a few of these things. And one of the things that I can note too is that, a trial from my perspective as a seed guy is the biggest no BS marketing approach. So it's, when I can get somebody out there and they can look at the flower, they can test it, they can see what it is. They're not just relying on me telling them, hey, this is a good seed to buy. They're able to see for themselves. So really getting ahead of the next year and looking at what varieties you're going to buy the year before is strongly suggested, and looking at trials, driving around and looking at other farms, and sharing your experiences with others. From here, I'm going to introduce Dan and Bob's section, and they're going to share with us some of the fascinating data that they found through their work at UC Davis. I'll let you guys take it from here. It might take a quick second because I'm going to have to transfer the screen share to Dan's screen. So if everybody can just bear with us for one moment.
Okay, I think I'm able to do this now.
Hey, Dan, you're ready to share your screen and let us all know what you guys have been getting into over there at UC Davis.
Okay, sounds good. I'm going to start off, and then we're going to do a little tag team here with Bob Hutmacher on some of the things that we've been studying over the last couple of years. The universities in general have not been able to do research on industrial hemp until 2019, when the laws changed, and so from that time we've dived into it primarily to try to understand a little bit more about the agronomy and the issues associated with industrial hemp. I have to say that our work has been primarily focused on CBD extract types of hemp, not on seed or other kinds of hemp that's grown for grain or other purposes. One of the things that I've done a lot of work in my career on new crops and alternative crops, and this is one of the reasons that we dived into hemp, because we'd like to see opportunities for farmers. And so one of the things that I always think about with new crops is that you got to think about it as a continuum between the production, the processing, the markets. The marketers have to consider all the way back to the production side, and those of us who are involved with production agriculture need to think about the entire continuum between production and markets. And as markets change, we need to perhaps change our production methods, including varieties and other kinds of practices. So I think this is an important model to really think through when we're understanding hemp, industrial hemp. Really a classic specialty crop with narrow markets and a possibility of rapid overproduction that we have seen in some years. And also we have bottlenecks that occur in different sectors. Some of the work that we've done at UC is we have a new hemp and cannabis center at UC Davis, including work in the med school. And those people have been working on chemistry actually for many years. Don Land and Department of Chemistry, and Gail Taylor and Plant Sciences has been working on a little bit on genetics and genomics. Charlie Brummer on breeding. We've had some people working on insects and diseases. Myself and Bob Hopmacher and Sarah Light and others have been working on agronomic work, and also work on analysis and sampling. In terms of the agronomic trials, which we'll talk about mainly today, we've worked with Kyogene over the last couple of years on plant density issues, and you can see here on the left, going from low densities to higher densities. Also on irrigation and water use studies started last year in 2020. We're going to continue those this year. In terms of the plant density studies, we worked with Kyogene with 2 varieties of their Autoflower types, and looked at these at both the Davis and West Side sites in Fresno County. These were on 60-inch beds with 30 inches between rows. Our target densities were starting off at about 7,000 plants per acre up to about 35,000 plants per acre. Remember, these are all Autoflower types. The shorter season as well as shorter height plants. So it's a good range of densities, but in some cases we were not able to obtain the right densities, but we did have a wide range of densities in our trials. This is some of the results. You can see here that yields were generally a lot better at the higher densities, up to about 30,000 plants per acre in the first year of study. The bud yields were only modestly improved at that rate, but they're still somewhat improved, up to about 2,500 pounds per acre at the West Side site, which is in Fresno County, and in Davis site, we had similar types of results, a little bit higher yields here. One of the things we should point out that the harvest index, which is the percentage of the plant which is harvestable economic product, if you look at the buds as the main product, is about 60 to 65% of the total biomass. And that's really different in the Autoflower types compared with full season types of hemp. Here are some of the results of the CBD content that we measured. You can see a little bit of a decline at high densities, really not that big of a difference of CBD at that level. And you can see, so we didn't really think that the density had that huge effect on concentration of the cannabinoids. However, if plant parts have a big, give you a big change between the bud and leaf and stem, as well as the whole plant, and this shouldn't be surprising to this audience. You have most of the cannabinoids are concentrated in the bud, but there are some in the leaves, a little bit in the stems. One of the things that was interesting about this with the 2 varieties, this one is the Maverick, and this one is I forgot the name of the second variety. If you could remind me of that.
Pipeline.
Pipeline.
Yeah, Pipeline. That's correct. And so you can see here that the whole plant CBD content is actually quite respectable, and that's largely because of the high percentage of colas in these Autoflower types. In the second year of the study, this shows you some additional data on this. The yields were higher in general in the second year, and these are the total biomass yields. So we're still waiting on some of the results from the concentration data, but you can see here the biomass yields are quite high at about 4,000 pounds per acre in the Maverick variety, quite a bit lower in the Pipeline variety. And again, you see this 60 to 70% harvest index that of the total biomass, we're producing quite a bit of bud on those crops. And the CBD concentration, again, is not really that dramatically affected. Actually, almost marginally, in fact, almost a different trend than in the first year, but I don't really put a lot of importance on this. I think that in general, density didn't have much effect on CBD concentration. We have to keep in mind the dramatic differences that we certainly observed compared with the full season types and the autoflower types. So you can see here some weeds coming in. When we plant these at too low of a density, that's going to be a bigger issue with especially these autoflower types. They don't form a full canopy if they're not at high enough density, and so that's one of the key problems with low density plantings. So in general, our conclusions here, we do need higher densities with these autoflower types. We would recommend direct seeding, I think, over transplanting, somewhere between 18,000 to 35,000 plants per acre, in that range. The planting pattern probably is pretty important. And in these trials, we haven't explored that in great amount, but certainly it's going to make a big difference in terms of canopy closure. Seed costs is most definitely going to be an issue, and of course,
I gotta
we have
chime in here on that one, Dan.
Yeah, well, we have we have breeders on the line, so they anyway. You want to make a comment on that? That'd be fine.
Yeah, sorry to interrupt. Yeah, thanks for putting that in your talk, because we look at 18 to 35,000 plants per acre, and the last thing we want to be looked at is like snake oil salesmen trying to push more seeds on you, with the with the bottom line not reflecting. So one of the things that we've done is looked at it in terms of a per seed cost with Autoflower, and we tried to rewind it and look at things on a more of a per acre cost, and that's how we develop our per seed costs. And we think our company is actually responsible for bringing the Autoflower seed price down quite a bit, because of the way that we looked at that and the way we saw it as a disadvantage for farmers, the way it was currently sitting.
Well, that's good. Yeah, and we look at the cost issues sort of independently, because they will change. They have changed actually over the last couple of years. So we're sort of looking at this independently of that and saying, look, we want to maximize CBD yields per unit area primarily, and that's one of the real objectives here. The other thing that comes into play here is the weed control issue, and is a grower going to use cultivation, and certainly we don't have an herbicide option at this point in time that's viable, at least legally. And also the other thing that comes into play is the harvest method. If something is hand harvested, it's a real different concept than if you're coming through with mechanical harvests or even harvesting the whole plant. So it's all of these things come into play. This year we're going to go with a different row configuration at 15 inches with about 30 to 35,000 plants per acre in our experimental plots for our nitrogen work and other work that we're doing this year. Any other ideas on this density trial comments?
No, that looks good to us.
Okay, so the other thing that we were really interested in is what's happening with the plant to plant variation. This is some data that Charlie Brummer's group has put together with their breeding work and where they are sampling large numbers of plants. And you can see here the relationship between CBD concentration and THC concentration. This gets at what the comments were made earlier. And it shows pretty clearly that if you get over about 11 to 12% CBD, the risk of those plants going hot is pretty substantial. There's 80% are square values. So the CBD concentration essentially explains about 80% of the variation in THC content in the varieties that we were looking at. And it could be that there's some varieties that don't obey that rule, but in general, there's a pretty tight relationship between those two. So that has a major effect on how we can, we basically, the two messages here are, one is that we have to pay attention to this CBD content and THC content over time. And also that we're not going to be able to, under current rules, maximize the CBD concentration necessarily in our crops. And it may be variety dependent, but I'm convinced at this point in time that virtually every variety has the capability of going hot, depending on the situation and of course when it's sampled and so forth. This is another graph that shows the degree of variation within, say, a variety. So these are families in the breeding plots, and they're a little bit more variable, but not that much more variable than regular varieties. And so even though the mean of these varieties is actually pretty much within the regulations, except for this one here, you can see the spread between individual plants and individual samples between, and this one is the mean of this group is about 0.26, which is under compliance. But then if some of those plants are less than half of that, and some are more than double. So the sampling issue is not a trivial point. And I think it's something we have to continue to pay attention to. We did some modeling on this, and if you take a look at any individual variety here, and you see here that even though the mean of that group of colas is acceptable, if you only took one sample or two samples or three samples, the degree of variation in that sample set is very, very high. It's just as likely that you'll be non-compliant as to be compliant if you only take one or two or three samples. So we would urge regulators and growers to make sure that you take a number of plants for your sample. So the message is here is that it's really a numbers game. I think even with fairly uniform varieties, you're going to see this type of curve, this type of degree of variation. And variation's a fact of life in biology. We accept that, and we need to recognize it. But there are some practical limits. We would probably recommend that we choose somewhere between 5 and 10 colas to represent a population, because 20 or 30 or 40 or 50 colas is going to be very, very hard for laboratories to process and represent on the subsampling side. So there's a lot of issues that need to be resolved there. So
Dan, quick question. Do you think that this will change the variance there, like this graph will change once the varieties become more stable? Because I know that you're running
it will
this
change to some degree, but we've done this type of study with even with hybrids, you sometimes see this degree of variation, not to as great a degree. Here's the other thing is that because this threshold is so low, right? That's one of the problems here, because the lower the number, the more variation makes a big difference. And that's part of the problem, because you have to take the biomass, which we're talking about pounds, right? You know, even if you take, say, 8 colas, you might have a half a pound to a pound or a couple of pounds. And then you have to grind that up. And so you have subsampling errors. So you have a whole series of errors that go into that. So you're always going to have variation. But the question is whether or not it's more limited. And we do have actually a whole group of samples on some of the Autoflower varieties from Kyogene, and we're going to be, we haven't had the data back from the lab yet, so we're hoping that we'll learn more about that. But
thank you so much. That's a really good insight.
Yeah, so I think all varieties are going to have similar types of curves like this, but with the better varieties, it'll probably be a little bit less variable. But as you know, anybody who's grown the crop, you know that it's somewhat indeterminate. So you have more mature colas, you have less mature colas, and so there's a whole series of colas that are becoming ripe or harvestable at different times. So I think variation is there, and we're going to need to educate. I think we have to educate the regulators too on this, because a lot of times the regulators are failing to understand this.
Yeah,
George,
that's a great point.
George, if I could just jump in here really quick. I can't say enough how much testing early and often really does help. It gives, and testing prices have gone down, getting a random sampling of your field that you can pull yourself. I was saying in an earlier conversation, I have just a kitchen food dehydrator that I got on Amazon. You know, I'll go out into my field or greenhouse or our indoor grows, and I'll go ahead and pull, you know, 5 to 7, maybe 10 different samples, you know, kind of in the mid range there. Dry them myself to save the drying cost money from the lab, and then let them homogenize the material for potency testing in weeks 4, 5, 6, and 7 of flower. It gives us just a wealth of data that we can really see when different cultivars, you know, mature. You know, and some of them end up maturing earlier than others, some later, you know, but testing early and often, it's not just good data for you, but it's also good for compliance. And if I could just take a quick thing, I would, I've seen everybody's questions in the chat. I'm more than happy to, I think you mentioned this, George, but I'm more than happy to go over, you know, whether it's the legislation, the testing, measurements of uncertainty, and total T compliance and cultivars going forward. So if everybody can just hang out till the end, I'd be happy to do that.
Thanks very much, Jason. Yeah, I think there's be a lot of interest in that. And I think Dan, are we going to move it along to Bob
Yeah.
here to close out the research section?
Here are the conclusions here. We got to look at both plant to plant variation and maturity and sampling methods. It's kind of a major issue. So with that, I'll move it on to the next talk, which I think Bob's going to talk about this. And Bob, just let me know when you want to go to the next slide.
Okay. All right. Thank you. Yeah, this is just some of the data that we have from an irrigation study that we're doing at Davis and Westside in cooperation with Oregon State cooperators who also have sites up at 3 different locations in Oregon. So this is 2020 data from Westside Research Center, and these are a couple of the non-Kyogene autoflower varieties that were included in the irrigation study that we did at Westside. And part of what this is just showing, we basically came up with an approach for irrigation that was kind of centered on water needs for full-size plants. And so one of the things that we learned certainly in the 2020 year was that despite the fact that we were aiming for 40%, 70%, or 100% of applications of water to meet evapotranspiration or crop water needs, basically we were over irrigating significantly when we were going with the 70 and the 100%. And you could kind of see that in terms of the plant responses here, maybe a little bit under irrigated on the 40% ET. In blue, that's the Kola yield showing in terms of a little more stress at that 40% ET level, and then really at the 70% and 100% ET non-stress plants, you didn't see large differences in Kola yield. You did see some increases with higher water in total dry weight. Next one. Okay, and this, by comparison, you look at the numbers for what we were for totals on the Autoflower, we were in the 4,000 plus pounds per acre type of total dry weight numbers. So these are, by comparison, the full season varieties, 2 that were included in this irrigation trial or multi-location irrigation trial. This is the San Joaquin Valley West Side location, and you can see in this particular case, when we went from 40% of ET to 70% intermediate water to 100% ET, you saw this stair-step type of response in total dry matter that was fairly significant, fairly large in amounts. And then you still saw down there in blue in the Kola dry weights, we probably peaked out at close to 4,000 pounds per acre. We did see a stair-step increase there in yield improvements as you went, but again, not as large an increase as you might expect in going from that 70% to the 100% ET. Next one. And one other thing we wanted to point out was we've had plenty of questions over the last couple of years about what we think are water use needs of the different types of industrial hemps that are out there are under consideration. And so what we're showing here is for the 2 cultivars of Autoflower, I came up with the averages for how much was applied water. And then that's the 3 bars on the left side at the 40%, 170% or 100% ET. And then on the right side is the averages across the 2 varieties for the full season types. So in looking at this, you can see that the applied amounts on the left for the average autoflower go from the low, the blue is applied water, and then so that goes from a low of about around 8 to a high of about 13.5 inches of applied water that we applied by irrigation. And then the amounts that are shown in that kind of reddish brown color, that represents extracted soil water, or water that was used during the course of the entire growing season. So what that means on the 40% AT is the combination of those two means about 10 inches of water for total crop water use in that low water application 40% ET treatment. The 70% ET treatment, the reason why those red and reddish orange or reddish brown color part of the bar is below the 0, what that means actually is that we over applied water greater than what the crop could use, and we ended up with a net addition or increase in soil water going from the beginning of the season to the end of the season. So what we were finding in general then is ET levels across the treatments for the autoflowers that range from about a little less than 10 inches to a high of about 13 inches. And I wanted to contrast that with what we saw with the full season. So for these varieties, with the full season, we're actually in the field waiting for harvest for about an additional 25 to 30 days longer than what was required for the autoflower readiness for harvest. And what that resulted in then is our lowest water application treatment, again, getting about 10 inches for the whole season of applied water in blue. That brown part on top of that is soil water extraction. So you can see in the full season types, we were actually using a substantial amount of stored soil water at the 40% and the 70% ET treatment. At the 100% ET treatment, we were only extracting maybe 1.5 inches or so. So we were probably coming pretty close to meeting full crop water requirements. And sorry for the long explanation, but the bottom line that's kind of important is that that means that these earlier maturing autoflower types, you're going to stop irrigating them earlier because they're going to be ready for harvest earlier. You're going to cut off probably somewhere in the neighborhood of maybe 25 or 30 days of additional irrigation timing, and even if you're not irrigating, crop water use for that extra 25 or 30 days. And that results in probably, I would say, somewhere in the neighborhood of about 5 to 7 inches less water use in the autoflower types than in the full season types. And in some settings, as we go into this, these drought conditions in the West and everything, that alternative uses for that extra 5, 6, 7 inches of water could be really important for some growers. Okay, the last couple things I wanted to mention was just that one of the advantages of being able to do variety trials and irrigation trials and so on is we've had plenty of chances to observe some of the pest and disease problems that have occurred. And we'll just mention that, we're not absolutely clear what the biggest problems would be, but again, in Davis and West Side, San Joaquin Valley locations, our worst problems were with worms that attacked and got up into the colas and did a fair amount of damage. Tobacco budworm and corn earworm were probably the predominant ones that we saw. We did also see leaf miners, ligas, mites, whiteflies, aphids. Didn't seem that those other pests actually caused that much damage or represented a particularly serious issue. We did see some disease issues, but predominant probably, at least in the San Joaquin Valley locations, was beet curlytop virus. And again, in all of these trials that we're doing, we're going to engage with the UC entomologists and pathologists to kind of make observations of what type of things are going on and maybe what to keep an eye out for. Okay, back to you, Dan.
Okay, well, thanks. And this brings it to a close. This year, we're going to be continuing with the irrigation trials and the nitrogen fertilizer, a new trial and nitrogen fertilizer response and uptake, which we are starting in about two to 3 weeks. And so that's what we're up to. Any questions? Is everybody still there?
Hey, Dan, yeah, so we wanted to leave the chat section for the very end here. So
Okay.
if we could move it back to
I'll stop sharing.
where we're at.
I think one of the questions was, did we see higher THC concentrations in water stress plants and weed? We've actually sent off all our samples to Oregon State, and they're not back yet. So we don't not know the answer to that at this point in time. So we'll try to get back to you. Hopefully those sample data will be coming back pretty shortly here. So we'll let you know.
You know, and the other thing I would add on that is that I think if people are going to, you know, frequently irrigate under drip, the plant response is going to be a smaller plant if you deficit irrigate, and that's different than what you might expect if you know, have like a hard shut off and stress the plant really hard. So again, the type of stress and the rate that the stress develops could be probably pretty important with this kind of a plant as well.
Thanks, guys. We're about to open it up for questions here via the chat column. I just wanted to really thank all the panelists and thank all the listeners. We appreciate you, and we're thrilled to be building the industry with everyone and collaborating with knowledge and hopefully good products back and forth to our partners up and down the supply chain. We're going to move to the next slide, which is our contact info. So you can all reach us here at these email addresses if you have any detailed questions beyond some of the brief questions that you're able to ask in the chat column. So I'll leave it on this page here as we go through questions. I'll open up the chat starting with some of the ones that came.
George,
So we got we got a question from Wayne Richmond. Are you aware of the potential smokable hemp ban in California, AB 45 and SB 235? And what are our thoughts? We are aware, and we're trying to help in any way possible. We have one of our colleagues, Matt McLean, is working at trying to push back on this with regulators. We highly recommend anybody listening to this in California that's interested in growing smokable hemp. We highly recommend that they reach out to the state legislature and provide their thoughts on AB 45 and SB 235. We think they're very unfair and we're not in support in any way. And we have another question from Kirk. Did anything change for MOUs testing? And I guess that's another California grower here. We were growing under MOUs and the testing there was different. And yes, there are no MOUs left in California. Everything is done through the CDFA now. And now the third question from Eli. Do you think that the focus on 0.3 total THC compliant cultivars is going to dramatically limit the available options and overall variety in terms of cannabinoid profiles and terpenes? Maybe that's a good question for Chris. Yeah.
So no, I don't think that it'll limit from a breeding perspective, because we can get all the same desirable consumer traits into any cannabinoid profile through breeding. So it's not going to at all limit the long-term options, but it'll definitely limit the short-term options for growers.
And then we have a question from Tim. Do you see, oh yeah, so you guys already answered that one, Dan and Bob. Oh, and just a comment from Wayne about the California ban on smokable hemp flower. Uh, he recommends that everyone reaches out to the California Hemp Association and joins their efforts in fighting the bad legislation here in California. Jason, do you have anything you'd like to add to any of the questions that were asked here?
Yeah, let me preface anything I'm about to say that I'm not a lawyer. I don't give legal advice, but I've been operating in the trenches of the hemp industry for the past 7 years. AB 45 is still nascent. It's undoubtedly going to face opposition. In addition to the California Hemp Association, we also work with, the Oregon Hemp Farmers Association, also hemp supporter, the Hemp Roundtable, and our legal representation is Harris Bricken. I don't see how AB 45 can include hemp and CBD products and exclude smokable flower when California was one of the first, medical and recreational marijuana, THC bearing cannabis programs in the country. It really makes no sense. But again, I'm not a lawyer. The second question for MOUs, I read that as a measurement of uncertainty. So maybe I couldn't answer that question because as far as I know, for measurements of uncertainty, they vary state by state. But I do have something to add on the question of focus for 0.3 total THC compliant. It's definitely going to, like Chris said, in the short term, limit available options. Now, there are, if you started what we did last year, focusing on total T compliance, you only had a couple of options, right? You had CBG, you had Autoflower, or you had a full photoperiod flower that could potentially grow in the high teens or 20s, and you had to early pull. Those options are still available. However, there are more and more. There are a couple right now. We just did a trial run of a couple of Kola Seeds, newer strains coming out. Happened to do them in a greenhouse, and you honestly, other than the fact that they were done in 75 days, you couldn't tell that they were Autoflower. They didn't have, but we've got mature soil beds in our greenhouses, so we give them the best living soil possible. But and I'm not paid to plug Kola Seed, but you really couldn't tell that these things were stunty. What I'm used to seeing from Autoflowers. Great terpene profile. We just harvested yesterday and today, so that is definitely a possibility. And as more, it's going to be a two-handed thing. As more people, more farmers, and more customers understand the regulations. I spent all last year fighting against other vendors who were still selling 20% CBD flower illegally, albeit, against my 7 to 10% CBD flower, and had to explain dozens, if not hundreds of times why. It's because I was the only one who was really following the law, as it was written and as it was made into law as of March 22nd. But again, I'm selling finished, dried, trimmed, post-harvest tested flower for the retail market. None of this stuff would have been an issue for me if I was just growing for extraction. So if you are growing for the flower market this year, I understand people may be hesitant to use Autoflowers. What I can tell you, I don't know, George, if you've released the names of the varietals that we test grew, and so I don't want to say them unless it's okay. But if you are considering growing and you care about growing for the flower market, and you've heard that Autoflower is not good for smokable flower market, I can guarantee you, based off of our trials, that there are multiple Autoflowers that will go total T compliant, and that I'm willing to put up on my website that Kola Seed offers right now.
Awesome. Thanks so much, Jason. I think there's another question here, and it's, is the video being recorded? And yes, we are recording this, and it'll be available on Tweedle Farms YouTube, Kola Seed YouTube, and we'll also send a link via email as a follow-up to all the participants here. So if you had any issues with audio or anything else, we will provide the edited recorded version. So unless there's anything else the panelists would like to add, I think there's actually somebody actually popped up in the Q&A here. I'm gonna take a look. That was the
There was one question from Alonso Bustos. What kind of opportunities are available for students looking into hemp research? And there are opportunities for students that are enrolled as undergrads. We did have a group this last year that did a cost study on hemp, industrial hemp, and also if funding was available for graduate students to do research in the field. So there is an opportunity there to answer that question.
And that's a great reason to email anyone on this list. If you guys have any further questions that we didn't get to, or if there's anything we missed or something that's a little bit more drawn out, just please don't hesitate to reach out. We're all in this together.